Swayam Prabha. Digital India. Educated India.
Hello everybody, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And in this course, so far we have discussed many aspects related to biotechnology as well as related to genetic engineering. So now today we are going to re-summarize or summarize the whatever we have learned so far in this course. So if you see, this course has 12 modules, and the content of this course is been divided into all these 12 modules. By now you might be very much familiar with this figure, which actually will be the core figure through which we were discussing the different aspects related to genetic engineering. So, we started with a very basic thing about understanding or about discussing the host, and then we discuss about the how you can isolate the gene from the host, and then you are going to cut these genes with the restriction enzymes, and then similarly we have also discussed about different types of plasmids, and then we have discussed about how you can do a restriction digestion, and that will actually going to generate the cohesive ends. So, you are going to have the cohesive ends onto the gene, you are going to have the cohesive end onto the plasmids, and then you are going to put these two components for the ligation reaction. That is how you are going to get the recombinant DNA, and then you are going to employ different types of DNA delivery methods to transform the bacteria, and that is how you are going to have the genetically modified organisms. And once you have this, you are actually going to do a screening process, so that you will be able to screen the transformed bacteria from the non-transformed bacteria, and ultimately once you got your desirable clone, this desirable clone can be used for protein productions. And subsequently to this, we have also discussed about how to purify the protein, how to break open the host cells, what are the different methods are available for break open the host cells, so that you can be able to release the content from the different storage compartment available within the host. And at the end, we have also discussed, once you got the product, how you can characterize that product utilizing the electrophoresis techniques, you are utilizing the chromatography techniques, and UV visible as well as the IR spectroscopy. And at the end, we have also discussed about the what is the application of genetic engineering or the biotechnology in different fields. For example, the animal sciences, the plant sciences, we have discussed about what is the application of the medical sciences, and we have also discussed about the application of genetic engineering in all different types of aspects related to the society. So, the content of this course is being divided into 12 different modules. The module 1 is going to deal with module 1 and 2 are dealing with the basics of the host. The module 3 and 4 are actually covering more about the basics of cloning 1 and 2. Module 5 and 6 are mostly been developing or dealing with the recombinant DNA technology, so that you can be, by utilizing all these, you can be able to generate the recombinant DNA. Once you generated the recombinant DNA, then the next step is that you actually going to overexpress. So, in the module 5 and 6, we have also discussed about different types of overexpression strategies, what you are going to use in different types of host strains, whether it is a bacterial strain or the yeast or bacteriophage or insect cell lines or mammalian cells. Then in the module 7 and 8, we have mainly focused on to the isolation and purification of the product, what you are going to overexpress in these host cells. And then module 9 and 10 is more on towards the characterization of the isolated product. So, in the module 9 and 10, we have discussed about the different types of the techniques what are available to characterize the product. For example, you can determine the molecular weight utilizing the gel filtration chromatography, electrophoresis, MALDI mass, or you can be able to determine the characteristic absorption or emission spectra of a molecule using the UV visible spectroscopy, or you can be able to run the IR spectroscopy to know what are the different functional groups are present in your compound. And then in the module 11, we have discussed about the different applications of genetic engineering as well as the biotechnology, which is for the social welfare. So, let us start from the module 1 and 2 and understand what we have discussed so far. This summary and the conclusion is very important, and very important because in due course you are going to face the exam of this course. So, we are just going to review the everything what we have discussed so far in a very fast forward mode, so that you will be able to refresh your memories, and if anything which is not going to be covered or which is actually not going to be understand, then you actually can go back to that particular module. You can again listen those video lectures, and that actually be the way you can be able to prepare yourself for the exam. The assignment what you are going to get in this particular week is actually probably a kind of a mock exam. If you do that, you are actually going to prepare yourself for the real exam, which is going to happen very soon. So, in the module 1 and 2, we have discussed about the basics of host, and what we have discussed, we have initially discussed about the introduction of biotechnology, how the biotechnology is helping the human society, and then we have discussed about the structure of prokaryotic cells, structure of eukaryotic cells. Then we have discussed about the differences between the prokaryotic and the eukaryotic cells. This is important because once you know the cellular structures of the prokaryotic as well as the eukaryotic cell, as well as the differences between them, you can be able to utilize or exploit the host strain, and you can be able to choose the perfect host strain what is required for overexpressing your protein or overexpressing your factors. Then we have also discussed about the difference between the plant and the animal cell, and then at the end in the lecture 3 we have also discussed about the isolation of different organelles or the fractionation strategies from the eukaryotic cell, as well as we have also discussed about the isolation of periplasmic fraction from the prokaryotic cells. So, we started very basic that with the very simple example of formation of bread, and that is a very classical example through which we have started with the course, saying that this is happening because people were making a dough, and then they were adding the very small quantity of yeast, and what will happen is that when the yeast is utilizing the glucose which is present in this dough, it is actually producing the carbon dioxide, and that carbon dioxide when it comes out from the dough, it actually makes the dough very fluffy, and that is how you are going to have a spongy texture of the bread, and that is what one of the classical example of the utilization of biotechnology for the social welfare. Then we moved on and talked about many of the important discoveries of the biotechnology in terms of the different eras, whether it is the pre-genomic era or the post-genomic era. Most of these discoveries were done simply because there will be a requirement of that particular type of product, and that is how the people have started doing the research as well as the different types of experiments, and that is how they have discovered many of these techniques or many of these products they have evolved. So, I am not going to discuss all these because this all we have discussed in the lecture 1. Then we have discussed about the structure of the bacterial cell, where we have said that the bacterial cell has the plasma membrane, bacterial cell has circular plasmids or circular genome, and then it has the extra chromosomal DNA that is called as the plasmid, and that plasmid is important for providing the resistance or different types of phenotype which actually are happening in a dynamic fashion, so that the bacteria is continuously exchanging their plasmids between the different bacteria present in the within the colony. And then you have the flagella. This flagella is actually important cellular structures of the bacterial cell, because it actually allows the bacterial cell to reach to the food particles, and it allows the bacteria to swim into the water or other kind of liquid media. And then we have the cytoplasm. So, the bacterial cell does not contain a proper nucleus or double walled nucleus, and in fact, there is no cellular organelle available which is double walled cellular organelle present in the prokaryotic cells such as bacteria. And bacteria has a very thick cell wall, and outer to this cell wall there is a capsule. This capsule is made up of polysaccharides, and this capsule is actually helping the bacteria to withstand the very high quality of the dehydration and all other kind of environmental stresses. Then we have also discussed about the composition of the cell wall, the composition of cell wall in the gram negative bacteria or the gram positive bacteria. The major difference is the presence of the peptidoglycan layer. So, what you see is a very thin layer of peptidoglycan which is present in the gram negative bacteria, whereas there is a thick layer of peptidoglycan in the gram positive bacteria, and that actually makes things very different between the gram negative versus gram positive. And because there is a difference in the thickness of the peptidoglycan layer, the people have discovered the stains which actually can be used to distinguish between the gram positive as well as the gram negative bacteria. So, what you see is there is a stain which is called as the gram stain, and the gram stain is being discovered by a scientist called as the Hans Christian Gram, and this technique is useful for differentiating the bacterial stain based on their cell wall composition, especially the composition of the peptidoglycan layer. And a detailed procedure anyway we have discussed when we were discussing about the bacterial cell wall as well as the utilization of the gram stains in differentiating the gram positive bacteria versus the gram negative bacteria. Then we moved on and we discussed about the eukaryotic cell. So, in the eukaryotic cell we have taken up the examples of animal cell as well as the plant cells. So, majority of the organelles are common between the animal cell as well as the plant cell, except that there are few differences. For example, in the plant cell it has a very thick cell wall which is completely absent in the animal cell. Compared to that, the animal cell has large quantity of mitochondria, because the animal cell is energetically very active compared to the plant cell. So, plant cell contains very low quantity of mitochondria. Similarly, there is a difference of the chloroplast. The plant cell contains the chloroplast, whereas the animal cell does not contain the chloroplast. And there are some more differences between the animal as well as the plant cell that also we have discussed. And then we have discussed about the plasma membrane, the composition of the plasma membrane. The plasma membrane is made up of two components. One is the lipid bilayer and the proteins which are present within the plasma membranes, and plasma membrane is a bilayer structures where the proteins are integrated. There are multiple types of proteins, the protein which are actually running all over the plasma membranes, or the protein which are either present onto the top layer or to the bottom layer. So, plasma membrane contains different types of receptors. This receptor could be for the food particles, this receptor for the capturing the signals, or they have many of the integral protein as well as the peripheral protein. So, what you see is a protein which is going through all over the plasma membrane, which means it is covering the both layer of the lipid bilayer, then that is called as the integral protein, whereas the protein which is present either onto the inner side or to the upper side, that is called as the peripheral proteins. The function of the plasma membrane is that the protein which are present onto the plasma membrane are working as a transporters. The classical example is the sodium potassium transporters. Then it also has the receptors. The receptor, as I said, is receptor could be for receiving the food particle. The classical example is the LDL receptor, which actually take up the LDL from the blood, and it actually does the recycling of the LDL receptor from the surface inside the cell, and so on. So, what happen is when the LDL is present, LDL goes and bind to the LDL receptor, and this LDL receptor containing LDL is goes inside, and that is how it delivers the LDL, and after the delivery it returns back to the plasma membrane, and that is how the receptor recycling is keep continuing into the cells. Then it has the enzymes, for example, the adenylate cyclase to catalyze the reactions. Then it has the receptors which are not participating into uptake of the food particles, but it is actually participating into the signal transductions. A classical example is the insulin receptor. If you remember, we have talked about that insulin which is flowing into the blood, when it goes and binds to the cells onto the which is when it binds to the insulin receptor present onto the cell, it actually drives a phosphorylation reactions downstream, and these downstream phosphorylation reactions actually changes the cellular metabolism in such a way that the glucose get converted into that the cell starts taking up the glucose from the external environment, and that is how that glucose is start converting into the glycogen, and it gets stored into those cells. When you require the energy, then the glycogen is going to be broken down and glucose is available for the cell to utilize into the metabolic reactions. Apart from that, it also provides as a support system. That support system is actually required for providing the protections, for communication to the other neighboring cells, and then it also allows the selective entry or the exit of substances within or outside the cell. Then it responds to the environment, and it also works in a recognition particle, which means the plasma membrane contains some of the receptors or some of the factors which actually been recognized by the cell. One of the classical example is the MSC class 2 or MSC class 1, which actually been recognized by the system or the immune system to say whether a particular cell is of the same person, which means whether the cell is of the self origin or the external origins. Then we have also discussed about the fractionation of the prokaryotic as well as the eukaryotic cells. Within the prokaryotic cell, we have discussed about how we can actually get the protein from the different compartments. So, suppose you are overexpressing the proteins, it can actually be present into the cytoplasm, or it could be present into the periplasmic fractions, or it could be present onto the outer membrane or the inner membrane. So, these are the different locations. You can have the protein onto the periplasmic fractions, outer membrane, it could be inner membrane. Some protein might be secretory, so it may go into the extracellular media, or the protein will be present into the cytoplasm. So, how you can actually fractionate the bacteria in such a way so that if your protein of interest is present into the periplasmic fractions, you could just isolate the periplasmic fraction, and that actually is going to help you in terms of the higher recovery of your protein, as well as it actually going to give you the better purification as well as the better yield at the end. And then we have also discussed about the fractionation of the eukaryotic cells. In that we have discussed how you can actually prepare the plasma membranes, mitochondria, chloroplast as well as the cytosol. And all this fractionation, during this discussion about the fractionation of eukaryotic cell, we have discussed about how you can use the density gradient centrifugations or the differential centrifugation techniques to achieve the targets, which means you can use the differential centrifugations to get the plasma membrane, mitochondria, cytosol or the chloroplast, whereas you can do the same thing by the differential centrifugation, which means you can actually centrifuge the particles at a different speeds, and that is how you are actually going to get the different particles at a different sedimentation coefficients. So, apart from that, once we were done with the in the module 1 with the basics of the host, then we moved on and understand the metabolism of the host, and in that context we have discussed about the metabolism which is operating within the prokaryotic cells, the metabolism which is operating within the animal cell, and the metabolism which is operating within the animal plant cells. So, we have discussed about the anabolic pathways, which means the biosynthetic pathway, or the catabolic pathway, which means the energy producing reactions. In this context, we have discussed about the glycolysis. So, glycolysis is the central pathway which actually started as soon as the glucose enter into the cytosol, and then it starts the glucose get channelized into the glycolysis with the help of an enzyme known as the hexokinase, and once the glucose get phosphorylated and produce the glucose 6 phosphate, then this glucose is going to be committed for the glucose metabolism, that is the glycolysis. Once this glucose 6 phosphate is being generated, it actually has no other option but to enter into the glycolysis, and then eventually it is going to produce the pyruvic acid. During this whole cycle, it is going to generate the NADH as well as the ATP molecules. So, we have also discussed about what will be the balance sheet of the ATP productions if you consider that the glucose is being utilized and glucose is going to be oxidized completely in the electron transport chains. So, what we will have done in the step 1 to 4, the two ATP molecules are being used. They are being used when you are producing the glucose 6 phosphate and as well as the fructose 1,6 bisphosphate. So, that time you have invested the two ATP molecule. After that, there is a generation of two molecule of glyceraldehyde 3 phosphates. So, below this whatever you see is going to be a split. So, glyceraldehyde 3 phosphate then will be get converted, and in the step 6 there will be a generation of NADH. So, each NADH in electron transport chain is going to generate the 3 ATP. So, there is a generation of 1 NADH, but because the two molecule of glyceraldehyde 3 phosphates are being generated, you are going to multiply this number by 2. So, this means molecules of NADH are going to be generated, and since each NADH in ETS is going to give you the 3 ATP molecules, ultimately it is going to give you 6 molecules. Then in step 7 you are going to generate 1 ATP molecule, which means you are going to generate 2 ATP molecules, and in the last step when the pyruvate is going to be generated, then you are going to generate another 2 ATP molecules. Ultimately, what will be the intake, or what will be the total production of glucose ATP? The total production of ATP is going to be 2. You have invested the 2 ATP molecules. So, the 2 molecules are going to be subtracted, which means the 8 is the net ATP which is going to be generated from the glycolysis. If the NADH, if NADH is going to go to the electron transport chain and it is going to be oxidized completely. Once the pyruvate is generated, it enters into the another cycle which is called as the Krebs cycles. So, pyruvate is going to be converted into the acetyl-CoA, and in this step you are going to generate 1 molecule of NADH. Then and remember that you are going to generate 2 molecules of pyruvate because you have generated the 2 molecules of glyceraldehyde 3 phosphate. And then acetyl-CoA will get channelized into the Krebs cycle to go with these intermediates, and as a result it is also going to generate large quantity of NADH, FADH2, ATP and GTP. So, if you see the balance sheet, what you are going to do, there is no investment of ATP as it was done in the glycolysis. So, glycolysis you are investing 2 ATP, so that the molecules were getting channelized into the carbohydrate metabolism, whereas in the Krebs cycle you are just simply using you are generating different intermediates, and that is how you are actually producing the different types of ATP molecule at different steps. In step 3 you are generating NADH molecule, then you are generating NADH molecule and so on, and ultimately what is happening, you are actually making the 15 ATP molecule from the one pyruvate which you are getting from the glycolysis. But since from glycolysis you are getting 2 ATP molecules, you are going to have the 30 molecules of ATP, total ATP generated after the Krebs cycle. So, if I ask you how many ATP are going to be produced during the carbohydrate metabolism, if you channelized one glucose molecule into the glycolysis followed by the carbohydrate, it is going to be 40, whereas the 2 molecule you are going to utilize. So, 38 is the net ATP gain after the 1 molecule of glucose oxidation, considering that the glucose is going to be oxidized completely. So, after understanding this, we have moved on and discussed about the anaerobic oxidations. So, you understand that the glycolysis or the Krebs cycles are operating because there is a plenty of oxygen available in the system, and that oxygen actually allows the electron transport chain to run. But in the absence of electron transport chain, you are going to have the anaerobic oxidations. So, if you have the oxygen present, then you are going to use the Krebs cycle or the TCA cycle, and that actually is going to generate the energy as well as the 3 carbon dioxide molecule. But if the oxygen is not present, then the pyruvate which is being generated after the glycolysis is going to be channelized and produce the lactic acid, or the pyruvate is going to be channelized to produce the acetaldehyde followed by the ethanol. And in the there are 2 enzymes which are actually working in this anaerobic oxidation. The enzyme 1 is called as the lactate dehydrogenase, and the enzyme 2 is called as the alcohol dehydrogenase, and these are the 2 enzymes, or under these conditions because of the anaerobic oxidations, you are actually, what you are doing is you are converting the pyruvate to lactate, or you are converting the pyruvate to ethanol. So, that has a long significance in terms of for the organism, as well as it also has a significance for the industrial applications. We have discussed all these, we have discussed about the reaction mechanism of both the reactions in detail in the in that particular lectures. So, if you would like to know the mechanism, you can actually follow those, you can follow that particular lecture again, and that will actually give you the details. So, what is the significance of this? For the cellular physiology, the anaerobic oxidation is that when the glucose is being utilized, it is actually producing the pyruvate, and in this process it is actually converting a 1 molecule of NADH to NADH. This NADH will be get converted into NAD plus only if the oxygen is present. But since the oxygen is not present, what will happen is this NADH is there will be no NAD plus present in the cell. If all the NAD plus get converted into NADH, only the NADH is going to be present, but there will be no NAD plus present. So, if that happens, there are so many reactions where the NAD plus is required, and that actually is going to stop the cellular metabolism. So, because of that, what will happen is that the pyruvate is going to be get converted into the lactate, and in that process the NADH is going to be utilized to produce the NAD plus. So, that is actually being done simply by running the anaerobic oxidation, which means it is actually a futile cycle where the glucose is getting converted into pyruvate, and the pyruvate is getting converted into lactate, and the in this process what will happen is the NADH is getting generated. So, this means it is actually making the pool of NAD plus available for the cell to run its cellular metabolism, which means to other reactions, not the glycolysis or the Krebs cycle, but all other reactions. So, that the cell will be sustaining for longer period of time, and in that process if the oxygen will be available on a later date, then the automatically this pathway is going to be cut down, and then the main pathway will be running, and then it starts producing the energy. What is the industrial relevance? Industrial relevance is that under these conditions, when the pyruvate is getting converted into ethanol, the pyruvate we are actually using this for producing the alcohol. And one of the classical example is that you can use the yeast as a molecule to run the alcohol productions, or the other example is the fermentation, where you are actually adding the yeast molecules, and because the dove has very limited quantity of oxygen, it is actually going to go through with the anaerobic oxidation, and under the anaerobic oxidations it is actually going to generate, and that is how it is going to give you the baking of the bread as well, because the pyruvate is getting converted into lactate. So, that actually is the example of where the milk is getting converted into the curd, because the lactic acid is going to reduce the pH of that particular milk, and in that process the milk is going to be get converted into the curd. So, this is what we have discussed so far in terms of basics of host. We have discussed about the cellular metabolism, we have discussed glycolysis, Krebs cycle, then we have discussed about the anaerobic oxidations, and at the end we have discussed about the significance of anaerobic oxidations. So, in the module 1 and 2 we have discussed more about the host, whether in terms of initially in terms of the cellular structures, and then subsequently in terms of the metabolism. But in the next module we have more discussed about the growth media. So, because once we understand the metabolism, you also should understand how to grow the bacteria or how to grow your host. So, for that then we have discussed about the different types of growth media, and the purpose of a growth media is to provide the protein, carbohydrate, lipids and the DNA. Why it is so? Because if you provide the amino acid or the building blocks, you can allow the organism to take up these molecules and generate the protein, carbohydrate, lipids as well as the DNA and RNA, and together all these components are going to facilitate the growth of that particular organisms. So, we have discussed different types of growth media for prokaryotic cells, for yeast, for mammalian cells, for insect cell lines and so on. In this particular type of module, we have also given you demos to how to prepare the bacterial culture media or the cell culture media, and what are the different precautions you should take while you are preparing these medias. Then we have also discussed about the growth kinetics of a typical bacterial growth kinetics, that basically we have lag phase, we discuss about the significance of log phase, stationary phase and as well as the death phase. So, this is all about the basics of the host, where we have discussed about the initially we have discussed about the cellular structures, followed by the metabolism and followed by the different types of growth media as well as the growth kinetics, and that actually will allow you to understand the potential of each host, how to grow that, how to manipulate and so on. And then once you know about the host, we moved on and try to give you the idea about the different types of the techniques, how you can be able to isolate a gene fragment under the next module, that is called as the basics of cloning. Because if you want to generate a recombinant DNA, you have to get the recombinant gene, or you have to get your gene of interest from the different sources. So, we discuss about the three sources, the sequence, the three conditions, the condition when the gene sequence is not known, or the sequence, or the condition when the gene sequence is known. When the gene sequence is not known, you either have the flexibility of getting these gene sequence from the genome by the help of generating a genomic library, or you can have the, if the protein is getting expressed, you can have the transcriptome, then you can actually generate the cDNA library. Ultimately, you are going to screen either of these libraries, and then you are going to get the clone containing your fragments, and that you can use for cloning reactions. Similarly, if the gene sequence is known, you have the gene sequence, then you prepare the primers, you get the polymerase chain reactions, that is going to give you the fragment, and that fragment you can be able to clone into the vector of your interest. So, the different steps what you have to go through with the construction of genomic library. So, in the step 1 you are going to do a isolation of genomic DNA. In the step 2 you are going to do a restriction digestion of genomic DNA. Step 3 you are going to do a purification of DNA fragments. Step 4 you are going to do a ligation of fragments into the YAC vectors, and then step 5 you are going to do a transformation into the yeast, and that actually is going to give you the genomic library into the yeast. Apart from that, for the cDNA library, first step is that you isolate and collect the messenger RNA, then with the help of the enzyme reverse transcriptase you produce the DNA or the cDNA, then you clone this cDNA into the plasmid or the bacteria, and then you do a transformation so that it will be taken up by the bacterial cell, and that is how you are going to get the cDNA library. So, these are the steps: isolation of messenger RNA, preparation of complementary DNA fragments, cloning into the suitable vector, and then transformation into the suitable host. Then we have also discussed about the different types of screening methods what are available. So, screening method can be worked onto the three criteria. One is called DNA sequence. So, DNA sequence property can be used to screen both the genomic library as well as the cDNA library. Then if the protein is getting expressed, then you can use the immunogenic epitopes with the help of the antibodies. So, this property can be partially useful to screen the genomic library due to the truncation of a full gene or no expression of a gene fragment, but this approach suits well to the screen cDNA clones. Then the third is enzymatic activity. This property exploits the ability of a protein fragment to exhibit enzymatic activity. It is useful for the screening of cDNA library, but not much for the genomic library, because again the same thing, because the protein may not express completely, because the small protein fragment might be present into the clone, or that particular fragment may not be giving you the protein or the enzyme. Apart from that, we have also discussed in detail about the PCR. We have discussed about how the PCR works, where the single template is being utilized by the help of the forward primer and reverse primer, and then you have the different steps where the annealing, denaturations and extensions, and that is how you are going to get the amplifications. So, you are going to have three steps. One is the denaturations. In the denaturation steps, the template is going to be broken down into the individual fragments. Then you have the annealing steps. Annealing step, your primer is going to bind. So, forward primer will bind into this, the reverse primer will bind into this, and then you have an elongation step where the primer, with the help of a primer and DNA polymerase, the next strand is going to be synthesized, and that is how, when if you started with the single molecule in the beginning, after the first cycle you are going to have the 2 molecules, second cycle you are going to have 4 molecules, and the third cycle you are going to have the 8 molecule, and so on. And if you would like to use or utilize or want to quantitate what is the amount of DNA what you are going to get, you can use this formula to calculate. We have also discussed about how to design the primers, whether it is the forward primer or the reverse primer. We have also given you a demo of the different types of problems what you are going to encounter, or how to actually validate the primer sequences, so that you will be able to understand whether you have designed a better primer or not. For example, you can be able to see whether the loop formation is there or not, or different types of secondary structures are forming or not, or whether the primer dimer is forming or not, and so on, and how you can be able to overcome these problems either by utilizing some of the reagents or chemicals, or you can be able to change the sequences in such a way so that it will still be able to amplify that particular region, but you will be able to overcome the formation of loops or the self ligations. Then polymerase chain reaction has multiple steps, as I said, initial denaturations, then you have the multiple cycles of denaturations, annealing, and then elongations, and that actually will continue for at least 30 cycles, and then you have the extra elongations, and then you have the final old at 4 degree, and that is how you are going to get the PCR done. And we have also discussed about how to analyze the PCR results, and how to know that whether the amplification is being done or not, and what are the different troubleshooting you have done to do. We have also discussed about how to set up the PCR reactions and so on. Then we have also discussed about the analysis of the PCR reactions. So, you can see that in a negative amplifications you are only going to see a template DNA, but if the amplification is done, then you are going to see the template DNA as well. So, if it is a negative amplifications, you are going to see a primer dimers, because the primer are not going to be utilized into the reactions, but if it is a positive reactions, you are going to see a amplified product, and the primer are mostly being utilized into the formation of that particular amplified DNA. Then we further moved on, and we discussed about the role of different types of enzyme in the molecular cloning. We have discussed about restriction enzymes, we have discussed about the polymerases, we have discussed about the alkaline phosphatases, and then we also discussed about the DNA helicase. Restriction enzyme is required for cutting the DNA at a specific site, polymerase is required for PCR amplifications, alkaline phosphatase is required for removing the terminal phosphate groups, and the ligase is required for joining of two DNA, so that you are going to get the recombinant DNA. So, restriction methylase system is like an immune system. It is actually helping the bacteria or the prokaryotic cells to understand whether the particular DNA belongs to them or to the other organisms. So, if it is belonging to other organism, it is not going to be methylated, whereas if it is belonging to them, it is going to be methylated, and that is how they have the restriction methylase system. So, in the restriction methylase system, the enzyme, for example, in this case the EcoR1 is going to go to the DNA and bind, and then it will look for whether the adenine which is bound is methylated or non-methylated. So, if the adenine is non-methylated, for example, in this case both the adenine molecules are unmethylated. So, in that case what will happen is that the enzyme will bind to this DNA, and then it is going to cut this DNA into the two fragments. Whereas, if the adenine is methylated, for example, in this particular DNA, the restriction enzyme will not be able to make the closed complex, because the methylation is on the present on the adenine group, and because of that the methylated DNA will not going to be cleaved by the EcoR1, and that is how there will be no DNA degradations, and there will be a DNA degradation if there will be no methylation. So, it is a kind of a protection mechanism exist within the prokaryotic cells. We have also discussed about the mechanism of the restriction methylase systems. So, in that particular thing what we have discussed is that suppose you have the different types of restriction sites RE1, RE2, RE3, RE4, and suppose you have added the recession enzyme. So, what will happen is the recession enzyme will go and bind to all these recession sites, but it will only going to make the closed complex when it will go and bind to its respective recession site. For example, in this case the RE3 is the recession sites for which the enzyme is being added, and so once it form the closed complex, it is actually going to cleave the DNA and going to generate the fragments. Once it cleaves the DNA, then the enzyme is going to be released. Then we have also compared the different types of recession enzymes present in the system. So, you have the type 1 enzymes, you have type 2 enzyme, and you have the type 3 enzymes. So, they are very different in terms of their properties, their cutting properties, their cleavage site as well as their recognition sites. So, for example, in the case of recognition sites, the type 1 it is non-palindromic, type 2 it is palindromic, and in the type 3 also it is non-palindromic. Whereas the cutting site is 1000 base pair away from the recognition sites. Type 2 it is within the recognition sites, and the type 3 it is 20 to 25 base pair away from the recognition sites, which means type 2 enzymes are actually going to, first of all, they are going to have the palindromic sites, and on the other hand they are also going to cleave the DNA within the recession sites. So, you actually know precisely where the enzyme is going to cut, whereas in the case of type 1 or type 3 they are going to cut away from where they are actually going to sit and bind. So, that is why the type 2 recession enzymes are preferred for the molecular cloning applications compared to the all type 1 or type 2 type 3. Then we have also discussed about how to set up the recession reactions. So, you are going to take the recession enzymes, you are going to add the BSA, and we have also discussed about that you are going to add the BSA only to reduce the star activity of these enzymes. Then we discuss about the ligase. Ligase is an enzyme which is actually joining the two DNA to generate the chimeric DNA, and that is actually the basis of the cloning. So, what you are going to do is you are going to cut the gene fragment which you have isolated from the genome, you are going to cut the vectors, and then you are going to cut them with the same set of recession enzymes, so that they are going to have the similar set of cohesive ends, and that is how you are going to put them into a ligation reactions. And that ligation reaction also we have discussed, and how to set up the ligation reactions, at what temperature you should incubate the ligation reactions, and what are the different precautions you should take while you are doing the ligation reactions. And then we have also discussed about the alkaline phosphatase. So, digested linear plasmid containing cohesive ends ends up on both sides with phosphate has a tendency to recircularize, which means if you have a fragment and you have the phosphate on both the ends, that actually has the tendency that these two actually will going to bind to the OH which are present onto the other side, and because of that, if you remove these phosphate groups with the help of the alkaline phosphatase, this fragment is not going to recircularize until it is going to get the other DNA. So, that is what it is shown here. If you have a circular plasmids which is actually going to be cut by an enzyme. So, you have a phosphate, you have a OH, you have a OH, you have a phosphate. So, if you put it for the ligation reaction, this plasmid is actually going to bind to each other to give you a circular plasmid instead of having an insert within that. So, if you treat this with an alkaline phosphatase, it is actually going to remove the phosphate which is present onto this particular plasmid, and what will happen is it is now going to have the OH. So, in that case what will happen is if you put the inserts, the insert phosphate group is there. So, it actually going to utilize the insert phosphate group, and as a result it is going to give you the recombinant DNA, whereas the OH is not going to be get recircularized and it will not going to give you the colonies. Subsequently, we discuss about the linker molecules, the significance of linker molecules, the utilization of linker molecules and so on. Then we discuss about the adapter molecule as well. So, in what conditions you use the linker molecules, in what condition you use the adapter molecules. Then in the molecular cloning we have also discussed about the vectors. So, the vector is the DNA which has a two responsibility, ability to carry the foreign DNA and the ability to replicate in host of your interest, which means it should have the ability or it should have the features, so that you could be able to integrate your foreign DNA, and then it should also have the feature to go into the host. This means you need the system for some machinery, so that it should have the ability to replicate within the host of your choice, and then it should be having the regions so which you can use to integrate your foreign DNA. So, what are the different criteria? The vector should be of low molecular weight, vector should be give you the phenotypic changes when it enters into the host, which means it either should give you the antibiotic resistance, it should give some color or something like that. Then it should have multiple cloning sites, so that you can be able to integrate the foreign DNA, and then it should be of high copy number, so that your desirable things should be more. So, what are the minimum component required to make a vector? You need origin of replication. So, origin of replication is required, so that the vector is going to replicate within the host. Origin of replication is going to be specific for that particular host. If you are preparing a vector for the bacterial system, then it should be for bacteria. If it is for the mammalian system, then it should have the mammalian system. If it is for the yeast, then it should be yeast. Similarly, it should have a selection marker, so that it should give the phenotypic changes, and those phenotypic changes can be selected afterwards, and then it should have a promoter. So, the promoter is a requirement for those plasmids where you would like to have the overexpression or the protein productions. Then we have all discussed about the different types of vectors. So, we have discussed about the bacterial plasmids, we have discussed about the phage-based vector, we have discussed about the yeast vector, and we have also discussed about the mammalian vectors. In all these classes, we have discussed about the inducible system and non-inducible system, which means we have discussed about the inducible system and we have discussed about the constitutive expression vectors. We have discussed about the yeast vectors, which are actually having the inducible promoter, so that you can use the inducers. Similarly, we have discussed about the inducible as well as the constitutive vectors in the case of mammalian cells as well. And then we have also discussed about the DNA delivery in host. So, we have this first initially we discussed about what is the philosophy of the uptake of the foreign DNA by the host, and later on we have discussed about the different types of methods or strategies available in different types of host systems. We discuss about the transformation preparation of competent cells to understand the transformations. So, for different bacteria the competing reagent is very different, and these competent cells are going to take up the foreign DNA and they will get transforms. And then we have discussed about how to prepare the competent cells. We have also given you a very small demo, so that you can be able to understand the different steps and different precautions what you should take while you are preparing the competent cells. And in a typical transformations, what you are going to do is you are going to first grow the bacterial cells, you are going to add the your plasmid of your choice. So, for example, in this case we are taking a plasmid which is ampicillin resistance, incubate them onto the ice, then you give them a heat shock at 42 degrees Celsius for 90 minutes, then you put them for recovery phase, and after that you do them plating. After the plating, you leave the cell plate into the incubator for 24 hours or 16 hours, and then that actually will going to give you the different types of E. coli colonies. Similarly, we have also discussed about the transformation in yeast. So, we have discussed about different types of methods of transformation available in yeast. One of the method is spiroplast method. So, in the spiroplast method, you first grow the yeast cells, then you are actually going to treat the yeast cells with the zymolase. That zymolase is going to remove the partially remove the cell walls, and that actually is going to generate the spiroplast, and then you add the DNA plasmid as well as the PEG to the spiroplast, and that is how the spiroplast will take up the DNA. Then you do the plating and incubate into the 30° for 4 days, and that actually is going to give you the transformed yeast cells. Subsequently, we have also discussed about the different delivery methods available in the mammalian cells. We have discussed about the chemical transfection method. We have discussed about the liposome or the lipoplex methods. We have also discussed about the bactofectin, and we have also discussed about the transductions. Individually also we have taken up many of these methods. We have given you the demo how to do the transfections into the bacterial cell, into the mammalian cells, what are the different steps you should follow. So, we have taken an example of liposome or lipoplex methods, and that is how we have actually shown you a demo to tell you that what are the different precautions you should take while you are doing the transfections into the mammalian cells. Then we have also, once you have done the transformation, you also have to screen the recombinant DNA. For screening of the recombinant DNA, you have the 3 choices. Either you go with the enzymatic activity which is going to be present. For example, in this case you can use the LAGZ as an enzyme. So, in the case of enzyme, you can have the insertional inactivations, which means if you clone within this enzyme, it is going to inactivate it. So, your untransformed one is going to give you the reactions, whereas the transformed ones are not going to give you the reactions. Similarly, you have the antibiotic resistance genes as a screening method, or you can have the phenotypic changes for such as the blue white screening. So, in the and then you have the complementation of the mutation, that is a typical technique or typical way of screening the things into the yeast. In the complementations, it could be a positive complementations. In the positive complementation, the host does not grow on the media lacking the functional gene, but the host transformed with the recombinant clone can be able to supply the gene product and required to grow in the media. So, complementation of media always been done onto the those strains which are actually growing onto a media which are deficient of that particular thing. So, once that deficiency is being provided by the transformation, it actually grows, and that is a positive selection. Whereas, in the case of negative selections, a chemical compound is added to the media which will be converted to the cytotoxic agent in the presence of gene product, and as a result it does not allow the growth of the wild type cell. But the host strain transformed with the recombinant clone has non-functional gene product and grows in the presence of compound in the media. Classical example is OMP decarboxylase, which actually is going to process the 5-fluoroorotic acid to a fluorodeoxyuridine, and the 5-fluorodeoxyuridine is a toxic compound. So, it is going to give you, it is going to it is going to kill the yeast cells, and that is how it is going to not going to form any colonies. But if you do a cloning within that gene, it is going to produce the non-functional OMP carboxylase, and that is how it is not going to catalyze this reaction, and it is going to give you the colonies. Then we have also discussed about the screening of transfected mammalian cells. We have discussed about the reporter gene assays which you can use to see whether the particular mammalian cell is being transfected or not, and ultimately we have also discussed about the conformation of the clone DNA. So, conformation of the clone DNA, that the cloning of that the recombinant clone what you got after the cloning is actually having the right sequence or the right nucleotide sequence. You can do that by simply sequencing the DNA. So, you have the 2 methods, dideoxy method or the Sanger methods, and that those 2 methods you can use to sequence the DNA, and that actually will give you the confirmation that the DNA what you are the clone what you have prepared is correct. So, after that we have also discussed about how to do the overexpression of a particular protein into the host. So, once you generated the transformed bacteria, you screen them, you are actually having the two possibilities. You have done the cloning into the cloning vector or into the expression vector. Cloning vector is always been used to answer the questions which are related to mechanism of transcription, replications, or it can be used for preparation of genomic or the cDNA library, whereas the expression vectors you can use the mechanism of translation or the protein production. So, ultimately if you clone it into the expression vector, you could be able to overexpress the protein into that particular host cells. We have discussed about what are the criteria to select a particular expression system. It depends on the quantity of the desired protein, it depends on the size of the particular protein, it depends on the compatibility between the source organism on the expression system, and then it also depends on the downstream application. So, considering these criteria, you can be able to choose the different types of host, whether it is bacterial host, yeast host, insects or the mammalian cells. So, in the host as a production machinery, we discussed different types of expression system, we discussed E. coli as an expression system, we discussed yeast as an expression system, we discussed the insect cell line as an expression system, and we discussed about the mammalian expression system. Within all these expression system, we have also discussed about how you can, because all these expression system have different types of promoters. So, they can have the different types of inducers which you can actually use to induce the protein production, and that is how they are very different. So, when you are when you are taking care of the E. coli or yeast, the manipulation methods are going to be very different from the when you are talking about the insect cell line or the mammalian cell lines. So, expression of a gene using the E. coli expression system is very simple. First you do the transformations, you get the transformed colonies, you inoculate few colonies into your media, let the media to grow, and then when it achieves the OD to 0.6, then you do the induction, you induce it with the 1 millimolar IPTG. It will actually going to start producing the protein. After that you centrifuge and check. So, you can see if you run it on the SDS page, what you see is in the control you do not have the bands for that particular overexpressed protein, whereas in the case of IPTG induced samples you have a protein of your interest produced by the bacteria. So, this is all about so far what we have discussed until the module 6, and in the subsequent lecture we are going to summarize what we have discussed from the module 6 to module 11, and in that particular process we are going to discuss about how to purify the protein, how to isolate, how to break open the host cells, and different strategies what we have discussed so far in this course, how to purify, what are different chromatography techniques what we have discussed, and ultimately we are also going to discuss few of the classical biotechnology applications, and that actually is going to refresh your memory, that is actually going to let you to understand few of the missed point what I might not have discussed while I was taking those lectures. So, I hope this kind of revision is going to helpful for you to understand and prepare for the your exams which you are going to face in due course. With this I would like to conclude our lecture here. Thank you.